and welcome to the journal webinar series of the American Society of Agronomy, Crop Science Society of America, and Soil Science Society of America. I'm Liz Gebhardt with Vadose Zone Journal. We're very glad you could join us today for Hydropedology, Soil, Water, and the Human Connection. Before we begin, I'd like to mention that questions can be entered in the Q&A section, and the moderator will address them at the end of the presentation. The webinar will be recorded, and the recording will be emailed to all attendees after the session. Now I'm happy to introduce our moderator for today's webinar, Dr. Thorsten Knappenberger, an associate editor for Vadose Zone Journal. Dr. Knappenberger is an associate professor of soil physics at Auburn University. He completed his BS, MS, and PhD at the University of Hohenheim, Germany. His research interests include transport processes in soils and sediments, irrigation efficiency and timing, spatial and temporal statistics, and stormwater remediation. And with that, Thorsten, I'll hand things over to you.
Thank you, Liz, for the introduction. So welcome, everybody. My name is Thorsten Knappenberger, and it's a pleasure today for me to introduce Dr. Johann Baumer. He's emeritus professor at the Wageningen University, and he will give a webinar on hydropedology, soil, water, and the human connection. Johann, please.
Well, thank you. Well, good morning, America. It's a pleasure for me to, and thank you for the invitation to make these remarks about hydropedology, soil, water, and the human connection, referring to the paper that is mentioned here on the slide. And I hope my remarks will inspire you to read the paper. Next. Next. Well, the basic premise of the story is that development, and that is what we all want, isn't it? Combining hydrological and pedological expertise in hydropedology is bound to be more effective than the two separate activities. Now, we know that hydrology is the study of seas, lakes, rivers, but also groundwater and water in the unsaturated Vadose zone of the soil, the latter studied by soil physics. And pedology is the study of natural soils as they occur in the field, studying physical, chemical, and biological properties. In hydropedology, focus on physical processes in the Vadose zone and on flow patterns of soil through soils in landscapes. Next. Now let's face it, soils without water are dead, and water without soils just flows away. So combining the two is a recipe for life. Let's realize that without defining overall clear goals, any activity may just drift away in all kinds of directions. That happens too often, I feel. So let's avoid this with hydropedology, and that's why we suggest to focus on sustainable development goals as defined by the United Nations in 2015 as our point at the horizon. And this is a logical kind of thing with, as I said, we're all in favor of sustainable development. Next. Now, these are the 17 sustainable development goals visualized in an easy manner. I'm not going into detail on it. I assume you're familiar with it. And the next slide, next, shows that the main issues covered by hydropedology are contributions to ecosystem services. And ecosystem services are services provided by ecosystems to mankind. And when looking at agriculture, we focus at healthy and vital crops and vegetation, SDGs 2 and 3, preservation of water quality, number 6, saving energy, number 7, mitigating climate change, number 13, and preserving biodiversity and soil health. That's a choice. You can even select more under certain circumstances, but for our part of the world, that is rather satisfying. Now, to achieve all this, land users, farmers, face an enormous challenge because they have to satisfy all of it. And it's like playing chess on 6 boards simultaneously. And the question is then, of course, how can we help them? Now, we can focus on hydropedological contributions to these services, and as soil scientists, focus on soil health as a guiding principle. And this includes, as a second step, soil chemistry and biology. That should be very clear, because soil is a living body. That is ultimately the point. And when we start with the water, then the nutrients, and then the the biology, it seems to be a logical sequence of looking at things. Next. Now, before continuing, I suggest we take a step back, because after more than 100 years of research, there is a staggering amount of abstract and clinical water and soil data ever more available in well-accessible databases. But just like people, water and soils have more dimensions than just their clinical data on texture, bulk density, and percent carbon, okay? And we should not lose sight of the fact that soils and water are alive components of ecosystems, just like man. We're all in this together. So let's therefore not forget about the importance of personal storytelling. That was the basis for our cultures for thousands of years before we were writing down things. And hydropedology certainly has stories to tell. Next. The biggest story of all for hydropedology, and I quote from the Bible, Genesis 2, verses 6 to 8, quote, "Water vapor rose from the earth and wetted its whole surface. God formed man from dust of the earth and blew life into his nostrils." That's a bewildering thought, isn't it? The hydropedology concept was right there at the start of it all. Now, many other religions link man to soil and to the land. And of course, to be practical, dry soil will not allow such a link. Man would just have crumbled in that case. Next. Now, a large number of methods is available to characterize water and soil. And there's a beautiful book series from the SSA, Methods of Soil Analysis, edited by Dane and Top, that is still quite relevant today. And back now, the soil moisture regimes that are crucial to assess the ecosystem surface as mentioned earlier. They can be measured, those moisture regimes, but also simulated with models based on Richard's flow theory, requiring moisture retention and hydraulic conductivity basic data, as we all know. But this flow theory implicitly assumes soil to be isotropic and homogeneous, and pedology shows that this is not so in nature out there. But there is no problem. This is not a negative talk, this is a positive talk, because there is no problem. We can handle this by modifying our methodology, by changing the boundary conditions of the system. And the papers I will have cited in my VZJ paper illustrate this specifically. So don't get me wrong, measuring and modeling of soil moisture regimes should still be a central and crucial activity. Now let's hear a few Dutch soil stories. Next. The first soil story is of a haplohumate. It's a sandy soil under heather acidification. The iron got mobilized and precipitated in the spodic horizon, a hard spodic horizon. And it was long unclear what the permeability was at that horizon. It was assumed that it was zero, but nobody had measured it because you could not put your rings into the hard cemented horizon. So we carved out large columns, measured the infiltration rate, and found that the infiltration rate was low, but was still there. And that changed the perception of the role of these soils in an ecological context. Next. The second story is of a Glossop valve. It's a soil formed in loess, silt loam, textile B horizon, leaching of clay, and in that B horizon there was preferential flow along the faces of the patch and leaching the clay from there. And on horizontal cut, you see on the right side, you see the dark red areas. These are the pads, and you see the light colored areas around them where the cracks were, where most of the water moved and removed the clay. Now, when you put cylinders here at random, you get an enormous variability of data. And when you measure the two populations separately, the inside of the pads and the cracks, you get two rather distinct populations. And that helps to understand the water flow in those type of soils. Next. Now, we introduced the bypass flow phenomenon that is important in structured soils with continuous macropores. And bypass flow is the downward flow of free water through continuous macropores. It can be cracks, it can be animal burrows through an unsaturated soil matrix. Next. Okay, we got to story number 3. This is a Levee soil next to the river, one of our best soils, loamy texture. And you see that the dark the vertical bands, these are worm channels. And a lot of them go to about 60 centimeters, where the more sandy subsoil starts that is partly still stratified. You see that also in the picture. And others go down as deep as 1.40 meters. Now, on the soil that had field capacity, we applied 2 showers, two regular showers, and measured the water content afterwards. The initial water content is the solid line on the right, whereas the moisture content with the dotted line is what we measured after the 2 showers. And you see wetting at the 60 centimeters and wetting at the 1.40 meters. And this was bypass flow, water running quickly into the worm channels and wetting the soil at the great depths. We also ran a sand model, assuming the soil was homogeneous, and then the wetting went only to about 4 centimeter depths. Quite a different story. Next. An extreme version of this, a heavy clay soil, a riverine clay. Heavy clay soil, you see large pads, and we did the same thing there. We measured bypass flow, and the solid line is what was calculated, the moisture content with the sand model, and the hatched areas is what we really measured. And we found a very great difference in the summer months, July through August. Showing that the surface soil was not wetted very much. A lot of the water went down into the cracks, and again, we could, by incorporating the bypass flow, we could predict the real situation rather closely. Next one. Now, in this particular case, water runs down the cracks, and we stain that, and you see these dark blue lines where free water is found on the faces of the beds. And you also see the roots there. So that creates a very special situation for water supply to the plants. Quite different from having water in a homogeneous root zone. And well, that could also be taken into account. Yes, next. Next, yeah. Now, bypass flow can be measured rather easily. The test is published in the SSA soil analysis book I mentioned before. And it can be incorporated in the models by changing the boundary conditions of the flow system. That can easily be done. Next. Now, just a little aside here, water will only flow into the macropores when the surrounding small pores at the surface of the soil matrix cannot handle the vertical flux of water. So soil with macropores form a highly democratic system, if you like. The little guys call the shots when determining bypass flow. I think that's a fascinating thought in passing. Okay, next. But there's more. Small standard core sizes don't work really well in structured soils. You need sample volume should vary as a function of soil structure. If you have a large patch, you should have larger samples to be representative. If you take very small standard cores in these highly structured soils, you get a lot of variation that you can avoid by having larger samples. And the second example here to the right is interesting in the sense that we had a very large sample. These are real measurements. And in that sample, the clay soil, there was a worm channel going down. And when it was attached to the subsoil, the K-set was 0.5 millimeter a day. When we detached it, this was 50 meters a day. And when we put a very light crust on top, that caused that the large pore was filled with air, but the matrix was still saturated. So in all cases, we had saturated soil, pressure 0, and gradient of 1 centimeter per centimeter. So the definition of K-set varied enormously. And it is important to see those morphological features to explain what might seem to be very odd measuring results if you measure out in the field. In that sense, it can be very helpful, but it can, of course, not replace the measurements. I want to emphasize that. Next. So the main lesson is look at the soil before you measure or model. Don't just pick some parameters from a database. That would be my advice. And remember, every soil has a unique story to tell. Next. The story 5 is somewhat different. It's an example from Oman, and it illustrates the difference between looking at versus seeing. Now, what they saw there is that there was local vegetation growth. That's the picture on the right side, on the bottom, on the right side. They found vegetation growth in a basin at some places and no growth at other places. and that could be traced back to local occurrence of silty layers containing more water. You see the picture of the complicated profile there on the left. That's where the vegetation was growing. There was pure sand where it wasn't growing. And this information was used to design, to characterize the system and to design innovative irrigation systems taking these processes into account. They have a very interesting cutting edge physical study work developed on that, which is referred to in my publication. And so again, it's a lot of people had looked at this, but nobody had seen things. And seeing things is one of the key points that we need, I think, in the profession. And of course, pedologists have always done that, being out in the field and looking at things. Yes, next. Back to the main story now. How about soil health as a guiding principle? The soil moisture regime is important for soil health. It's one of the indicators, if you like, and it's defined as the capacity of soils to contribute to ecosystem services in line with the SDGs. Soil health requires additional indicators and thresholds for soil structure, soil fertility, soil pollutants, and soil biology. And of course, there's no time to go into detail on that, but you need them all. And one procedure is that to say soil health is good when a soil satisfies the 5 thresholds. Next. Now, of course, too much water isn't any good. Next. And R is too little. Next. Now, I think it's interesting to compare the soil health assessment with a medical doctor taking temperature, blood pressure, and other indicators for human health. Yeah, we have these indicators for soil. This is what we have for humans. Only when all tests pass the threshold, values can be, then the healthy verdict can be passed. And this is true for both man and for soils. Now, soil quality, and the term is often used, mixed up a little bit with soil health, which is confusing for outsiders. Soil quality can then express the range of soil health values for any given soil type. Compare this with health data for certain cohorts of people, as they call it. Health data for 18 to 25-year-olds, 80-plus-year-olds, males versus females, etc., etc. In this analogy, a soil type compares with a health cohort, and this acknowledges the particular character of each category of patients or soils. Never require an 18-year-old to perform like an 18-year-old, or a sandy soil to act like a clay. And again, every soil has a unique story to tell. Yes, next. So far, so good, but what about the real world out there? Now, 2 aspects are important. First of all, soil and water in a regional context, because I talked about soils now, particularly in terms of soil profiles, and 2, making sure that hydropedology becomes part of environmental rules and regulations related to land use. And our South African colleagues show us the way by studying regional water regimes with hydropedological approaches and digital soil mapping. I refer to that in the paper. Please read it, because these are important papers. They have actively taken part in the lawmaking process, so there are now land use planning laws requiring a hydropedological analysis. Next. And they show, this is one of the beautiful things they show this landscape diagram where water movement is very complex. It infiltrates, it runs over the surface, it runs over subsurface horizons and all that. And all the profiles you see here are can be used to derive those processes, because those processes leave very clear marks in the profile. That's really a beautiful example that can qualify as story number 6 in this particular presentation. And the next slide shows a theoretical landscape flow model, assuming homogeneity, and it shows, of course, a quite different pattern. Next. That leads me to my final remark. Hydropedologists of the world unite. There is important work to do. Thank you very much for your attention.
Thank you, Johan, for your interesting presentation. We have a few questions in the chat that we can go over. So we have about 10 minutes left for questions and answers. So let's maybe start with this one. can you discuss representative elementary volumes and landscapes to capture variability?
The representative elementary volumes, well, in terms of the soil profile, you have to look at when you have pedal soils, you have to look at the size of the pads, and the rule of thumb is that we did joint work with Cornell University years ago. You need about 20 elementary units of volume for a sample to be truly representative of the soil you want to characterize. And that's basically the rule of thumb that you have, and that means that in different soils you get different volumes, and using very small samples can result in a high variability, which makes interpretations a bit more difficult.
Okay. Then we have another question. How does hydropedology contribute to our understanding of contaminant transport and remediation in soil and groundwater systems?
Well, that's an important question, because we find pollutants sometimes in groundwater that shouldn't be there according to the models if we assume soil to be isotropic and homogeneous. Even if you know you use moisture retention and hydraulic conductivity data determined by the standard methodology, if you assume that everything moves through the entire soil matrix and you have a clayey soil, you have a lot of absorption capacity, so nothing will reach the groundwater. But if you have bypass flow, that's one of the main reasons why looking at bypass flow is important. A lot of pollutants may move very rapidly down to the groundwater. This is what's happening, and then it's good to recognize that. So in that sense, the pedology describes one of the things that happens in pedology was describing soil structure and the porosity. You can functionally characterize that with staining. That helps a lot too. If you have this downward flow, be careful, because then it could mean a rapid distribution of excess nutrients or pollutants to the groundwater. Of course, also the infiltration works very well in terms of looking at surface flow, land overland flow going to surface waters. That's where the macropores work well, because they catch more water, so more water can be absorbed. So you have to balance these two things, but and of course you can interrupt the continuity of the macropores by shallow tillage, for example. When the pores are not continuous, then the water will not move into the macropores. And think of my example of the large soil core with the one continuous pore with a very light crust on top. Then the water moves only through the matrix and not through the macropores, and then all the absorptive capacity of the soil is used, so to speak, and avoids the pollution of the groundwater. Yep.
Okay, then we have another question. I appreciate the emphasis on morphology working together with soil physics. How much morphological detail would be useful, in your opinion, to assess soil health?
How much detail?
How much morphology do we need to know to understand soil health?
That's a good question. Well, that's quite a jump from some morphology to soil health, of course. But let me put it that way. Soil health, we have the various elements of soil health, and soil moisture regime is one of them, but the others are there too. And the processes in the soil are not, the main point I think of what we've been trying to say, are not always homogeneous in the soil. So you may have bypass flow, you may have preferential flow along the larger pores, and then the matrix, the main body of the soil, does not participate in the processes, and that has an effect on the water regime, as we have discussed here, but it has a lot of effect on the fertility dynamics of the soil. You may have excess leaching. It has effect also on the biology, because the soil will remain drier when the water moves down than it would have been if the water would have percolated very well here. And it has certainly an effect on the carbon dynamics. The carbon decomposition, the carbon dynamics, so to speak. The whole moisture content of the soil has a big effect on that, and having the morphological effects of the varied water movement, not as a front, but separately, has a clear effect on that. So in that sense, it affects all the various indicators for soil health, and that's why it's important to consider it when you try to find a unified value for the soil health.
Okay. Then we have another question that goes towards sampling. We are using intact soil cores to understand drought response in wheat crop. Based on your experience, what diameter soil core would represent the soil in the field?
Okay, good point. Depends on the soil. You cannot have a standard core size if you want to get real representative data. So if you have a structure with relatively large structural element, PEDs, then you need a relatively large sample in order to be representative. If you have smaller elements, it can be smaller. If you have a sand, usually the 300 cubic centimeter cores are excellent. That's no problem. That works well. In Holland, we often use 100 cubic centimeter cores, and that can work in the sand as well. So it depends on your structure. And the basic message is that before you put in the cores or you sample, take a good look at the soil. See what it is like. In terms of macropores, are there cracks, are there worm channels, are there continuous root channels, what are the size of the pads, are there pads, and this is all part of the standard soil morphological description. But you need to make the measurements. I've made that point. Just describing it is not enough. That it's a step towards making the measurements more representative. And if you do that, the statistics you're applying in order to interpret your results will also be easier, because your standard deviation is considerably lower if you have representative samples. Because if you take unrepresentative samples, you get a very high standard deviation, and then it becomes very difficult to make a judgment as to the fact whether a certain treatment is significant or not. So in that sense, I think it is also important to consider these kind of things. So look before you sample. I guess that was one of the points I made, but looking alone is not going to do the job. Let's be clear on that as well.
Okay, and then we'll take that knowledge of variability a step further and take it into modeling. Here's a question. How can theoretical models still be effective if the subsoil varies greatly within a land?
It's a very good point. We describe one soil type, but we know that there may be a lot of variation within the field. We've been involved with precision farming, where this was a point, and then it is necessary to study the heterogeneity and then find out in what way you can generalize it in a way that is still meaningful to express this variation. And that's where also remote sensing can help a great deal, by taking images of the surface and find out how the crop reacts on those differences, because all the differences we see in terms of texture, bands, and what have you, does not necessarily always translate into reactions by plants. So it helps then, if it is a very heterogeneous soil situation, to look at remote sensing pictures and see what the reaction of plants is to the variation. And we often find that then larger areas can be distinguished that somehow seem to react in a comparable manner. But this is a point. One way also to deal with that is to make point observations and use geostatistical techniques and digital mapping techniques to derive patterns from that. And it is not sufficient to do the classical soil survey procedure of making a few augerings and draw some lines on the basis of sometimes rather qualitative legend of the soil map. So you need then more modern techniques, be it remote sensing or be it point observations and geostatistics and digital mapping, in order to find patterns. But this is a very important question, because there are some soil types that are very heterogeneous, and then it's rather difficult. There are other soil types that are more homogeneous, and then it is easier, but can be done in both cases. But with heterogeneous soils, it is indeed much more difficult. Very true.
Okay, and then we have one final question or comment. So any thoughts on future research priorities? Where are we going from here?
Well, that's a big one, and I want to be a little modest here. You know, my goodness, we've done quite a bit of work on soil health, and one thing, to make a long story short, is to say we need indicators, and I mentioned that those 5 I mentioned there, okay, and I've also mentioned the ecosystem services for the entire farming system. Now, looking at that, these indicators are quite reasonably to be established. When you talk about soil structure, we have both density, penetration, resistance, and all that kind of stuff. What is very much needed are the thresholds, the separation between what is good and what is not good enough. I put it in a positive way, okay? I'm not saying good and bad. So what are critical values? What are, for a given soil in a given region, what are critical bulk densities? What are critical penetration resistances for the roots to penetrate? What are critical organic carbon contents? Very important for climate change and also for soil behavior. If you have to say what is good and what is not yet good enough, I think the major thrust should be on threshold values, because we have still not enough of that. For groundwater, we have 50 milligrams liter nitrate, 50 milligrams nitrate per liter. That has been there for decades, but we don't have comparable things in soils, and we need that very much in order to make a link with legislation and rules and regulations that are transparent and also effective to communicate with practice and to be really effective in getting good results.
Okay, so we passed the 30-minute time for this webinar. Johan, I want to thank you very much for taking the time today and explain all this to us. And I want to close with a comment of an attendee who was writing, "Thanks for allowing a lecture of one of my former professors at WU 30 years later."
Okay. I'll find out who it is. Thank you very much.